body's calcium level stays stable thanks to three hormones: parathyroid hormone, vitamin D, and calcitonin. Parathyroid hormone and vitamin D help increase calcium levels, whereas calcitonin helps to lower them. Now let's focus on the role of calcitonin. The majority of the extracellular calcium, which is the calcium in the blood and interstitium, is split almost equally into calcium that's diffusible and calcium that's not diffusible. Diffusible calcium is small enough to diffuse across cell membranes, and there are two subcategories. The first is free ionized calcium, which is involved in all sorts of cellular processes like neuronal action potentials, contraction of skeletal, smooth and cardiac muscle, hormone secretion, and blood coagulation, all of which are tightly regulated by enzymes and hormones. The second category is complexed calcium, which is where the positively charged calcium is ionically linked to tiny negatively charged molecules like oxalate and phosphate, which are small anions that are found in our blood. The complexed calcium forms a molecule that's electrically neutral, but unlike free ionized calcium, it's not useful for cellular processes. Finally, there's the non-diffusible calcium, which is bound to large negatively charged proteins like albumin. The resulting protein-calcium complex is too large and charged to cross membranes, so the non-diffusible calcium is also uninvolved in cellular processes. Now, calcitonin is a polypeptide hormone involved in regulating blood calcium levels. Calcitonin comes from the parafollicular cells, or C cells, of the thyroid gland, which is a gland located in the neck that looks like two thumbs hooked together in the shape of a V. The thyroid gland is made up of thousands of follicles, which are small spheres lined with follicular cells. C cells are adjacent to follicles, more precisely in the connective tissue that separates the follicles. C cells synthesize pre-procalcitonin, a peptide with 141 amino acids, which becomes procalcitonin after a single peptide is cut off by an enzyme via proteolytic cleavage, leaving 116 amino acids. From there, procalcitonin is cleaved again to the 33 amino acid long immature calcitonin, and finally cleaved one more time into mature calcitonin, which has 32 amino acids. It's then stored in secretory granules in C cells waiting to be released. Normally, total blood calcium is between 8.5 to 10 mg per deciliter, and changes in calcium levels are detected by calcium-sensing surface receptors on C cells. If the concentration of calcium in the blood goes above the normal range, C cells release calcitonin, lowering blood calcium levels. What makes calcitonin different from, say, parathyroid hormone in calcium homeostasis is that it's not actually involved in minute-to-minute regulation of blood calcium concentration. In fact, the exact role of calcitonin in the body is still a bit of a mystery, because if the body stops producing calcitonin, for example when the thyroid is surgically removed, the regulation of blood calcium doesn't seem to be affected. Weird, right? Still, we do know that the body responds to calcitonin, and it's sometimes used in high doses to treat hypercalcemia. Calcitonin contributes to lowering blood calcium levels in two ways. First, its major action is in the bones, and second, through minor effects in the kidneys. In the bones, calcitonin binds to the calcitonin receptor on the basal surface of osteoclasts, which are the bone-eating cells. Normally, osteoclasts attach to the bone matrix, and its membrane forms many little arms called a ruffled border, which helps them to attach to the bone, and increased surface area through which they secrete acid that breaks down the bone, which is also called bone resorption. This releases the two minerals that make up bone, calcium and phosphate, into the blood. When calcitonin binds to its G-protein coupled receptor on osteoclasts, a G-protein is released inside the cell, which activates adenylate cyclase. This enzyme converts adenosine triphosphate, or ATP, into cyclic AMP, or CAMP. Increased levels of CAMP cause the osteoclast to decrease the number of arms formed by the membrane, and they can't maintain a ruffled border, leading to decreased bone resorption and decreased blood calcium concentration. Second, calcitonin has a minor effect in the kidneys, more specifically the distal convoluted tubule, which also happen to have calcitonin receptors. When it binds to the receptor, it decreases calcium and phosphate reabsorption by the principal cells of the distal convoluted tubules, which use sodium-calcium channels on their apical surface to return calcium from the urine back to the body. So, increased calcitonin leads to more calcium and phosphate being lost in the urine, also known as calciuria and phosphaturia. Okay, so as a quick recap, Calcitonin is released when the calcium sensing receptor on a parafollicular, or C cell, detects an increased level in extracellular calcium. It then works to inhibit bone resorption and causes phosphaturia and calciuria by the kidneys, decreasing the level of extracellular calcium. These effects help to keep the total blood calcium levels between 8.5 to 10 mg per deciliter. The body's calcium levels stay stable thanks to three hormones: parathyroid hormone, vitamin D, and calcitonin. Parathyroid hormone and vitamin D help to increase calcium levels, whereas calcitonin helps to lower them. Let's focus on the role of parathyroid hormone. The majority of the extracellular calcium, which is the calcium in the blood and interstitium, is split almost equally into calcium that's diffusible and calcium that's not diffusible. Diffusible calcium is small enough to diffuse across cell membranes, and there are two subcategories. The first is free ionized calcium, which is involved in all sorts of cellular processes like neuronal action potentials, contraction of skeletal, smooth and cardiac muscle, hormone secretion, and blood coagulation, all of which are tightly regulated by enzymes and hormones. The second category is complexed calcium, which is where the positively charged calcium is ionically linked to tiny negatively charged molecules like oxalate and phosphate, which are small anions that are found in our blood. The complexed calcium forms a molecule that's electrically neutral, but unlike free ionized calcium, it's not useful for cellular processes. Finally, there's the non-diffusible calcium, which is bound to large negatively charged proteins like albumin. The resulting protein-calcium complex is too large and charged to cross membranes, so the non-diffusible calcium is also uninvolved in cellular processes. Now, blood calcium is regulated mainly by parathyroid hormone. Parathyroid hormone, or PTH, comes from the parathyroid glands, which are four pea-sized glands buried within the posterior part of the thyroid gland. Inside the parathyroid glands are the parathyroid or chief cells, which synthesize a protein called pre-pro-PTH in their endoplasmic reticulum. This long protein chain is 115 amino acids long and contains the parathyroid hormone segment, but also a pre-segment and a pro-segment. In the endoplasmic reticulum, an enzyme called signal peptidase cleaves the 25 amino acid long pre-segment off. Then, the pro-PTH is then moved to the Golgi apparatus, where a trypsin-like enzyme cleaves off the 6 amino acid pro-segment, resulting in the final parathyroid hormone, a single-chain polypeptide with 84 amino acids. At that point, the parathyroid hormone goes from the Golgi apparatus into a secretory vesicle within the cytosol, where it waits to be released. Normally, total blood calcium is between 8.5 to 10 mg per deciliter. Changes in calcium levels are detected by a surface receptor on parathyroid cells called the calcium sensing receptor. This calcium sensing receptor is connected by a G protein on the intracellular side of the cell membrane to an enzyme called phospholipase C. When the extracellular calcium level is normal or higher than normal, lots of calcium binds to the receptor, which activates phospholipase C, or PLC. Phospholipase C then splits phosphatidyl inositol bisphosphate, or PIP2, into two molecules, diacylglycerol, or DAG, and inositol triphosphate, or IP3. Then, IP3 diffuses through the cytoplasm to get to the endoplasmic reticulum, where it binds to a receptor called inositol triphosphate receptor, or INS3PR, on a ligand-gated calcium channel. This opens the channel, and the calcium that's being stored in the endoplasmic reticulum is released into the cytoplasm, making intracellular calcium levels increase. High intracellular calcium concentration stops the secretory granules that hold PTH from binding to the chief cell's membrane. Now, when there's less calcium in the blood, then the opposite chain of events occurs. Less extracellular calcium binds to the receptor, which means calcium remains in the endoplasmic reticulum, and the secretory granules bind to the cell membrane and PTH gets released. Parathyroid hormone works to increase extracellular calcium in three ways. First, in the bones, PTH binds to receptors on osteoblasts, which are the bone-building cells, and gets them to release cytokines. These cytokines include receptor activator of nuclear factor kappa B ligand, or RANKL for short, and macrophage colony stimulating factor, or MCSF. These cytokines help get multiple macrophage precursors to fuse together and form a single osteoclast, which are the bone-eating cells. This is kind of like how 5 robot lions join together to form a single Voltron. Osteoclasts break down bone, and the 2 minerals that make up bone, calcium and phosphate, are released into the blood. But in the blood, phosphate binds to calciums to form complex calcium, which can't be used for cellular processes. To stop this from happening, a second thing that parathyroid hormone will do is to bind receptors on the tubular cells of the kidney's proximal convoluted tubules. This stops the sodium and phosphate cotransporters on the apical surface of the tubular cells from reabsorbing phosphate from the urine. As a result, phosphate gets lost in the urine and it's called phosphaturia. Parathyroid hormone also binds to the principal cells of the distal convoluted tubule. This causes tubular cells to start making more sodium-calcium channels, which get embedded on their apical surface and increases reabsorption of calcium from the urine. Third, parathyroid hormone helps the body convert the precursor of vitamin D, called cholecalciferol, or D3, into its active form, vitamin D. Cholecalciferol is synthesized by keratinocyte cells in the epidermal layer of the skin when it's exposed to sunlight, but it can also come from food. Regardless of the source, cholecalciferol travels to the liver, where the enzyme 25-hydroxylase converts it into 25-hydroxycholecalciferol, or calcidiol. Calcidiol then travels to the proximal tubular cells of the kidneys, where the enzyme 1-alpha-hydroxylase, which is also activated by PTH, turns into 1,25-dehydroxycholecalciferol, also known as calcitriol, or active vitamin D. Active vitamin D then goes on to the gastrointestinal tract, where it enters the enterocytes of the small intestine and increases the activities of the calcium channels on the cell membrane, allowing them to absorb more calcium from the food. Alright, as a quick recap, parathyroid hormone is released when the calcium-sensing receptor on parathyroid cells detects a decrease in extracellular calcium. It then works to increase the level of extracellular calcium by stimulating bone resorption, causing phosphaturia and renal calcium reabsorption, as well as calcium absorption in the digestive tract, and altogether, these effects help to keep the extracellular levels of calcium between 8.5 to 10 mg per deciliter.
Phosphate is a negative ion, noted as PO4, while calcium, noted as Ca, and magnesium, noted as Mg, are positive ions. Now, about 85% of phosphate, along with 99% of calcium and about 60% of magnesium, are located in the bone matrix. Phosphate and calcium combine to form calcium phosphate, which makes up the hard bone matrix of bones and teeth, and magnesium helps strengthen it. Okay, now let's start talking specifics about phosphate. The remaining 15% of phosphate is found almost entirely in the intracellular fluid, or ICF, while only less than 0.5% is found in the extracellular fluid, or ECF. Now, most of the ECF is made up of plasma, and in the plasma, 90% of phosphate circulates free, while 10% is bound to plasma proteins. Phosphate plasma levels range between 2.5 and 4.5 milligrams per deciliter. Phosphate is a component of nucleotides that make up the DNA and RNA, high-energy molecules like adenosine triphosphate, and metabolic intermediates. Phosphate also acts as a buffer for hydrogen. Okay, now phosphate comes from our diet, and the daily recommended phosphate intake is about 1 gram per day, and we can get it from chicken, turkey, or pork. Once ingested, phosphate is absorbed in the GI tract, into the bloodstream, and then it goes where it's needed, such as the bone, and the rest is excreted. Okay, let's see how phosphate is handled by the kidneys. See, the kidneys are made up of lots and lots of nephrons, and each nephron is made up of a renal corpuscle and a renal tubule. The renal corpuscle, in turn, is made up of the glomerulus, which is a tiny clump of capillaries, and Bowman's capsule surrounding it. So, blood gets to the glomerulus through the afferent arteriole, which is a branch of the renal artery, and leaves the glomerulus through the efferent arterioles. These vessels act like a coffee filter, allowing everything but red blood cells and proteins to pass from the bloodstream into Bowman's capsule, which is connected to the renal tubule, and the resulting fluid is called filtrate. Now, upon exiting the glomerulus, the efferent arterioles divide into capillaries a second time, forming the peritubular vessels, which wrap around the segments of the renal tubule, the proximal convoluted tubule, the U-shaped loop of Henle, which has a descending and ascending limb, the distal convoluted tubule, and the collecting duct. As filtrate passes through the renal tubule, ions like phosphate, calcium, and magnesium are filtered from the capillaries into the lumen of the tubule, and reabsorbed from the lumen into the capillaries, depending on the amount of sodium in the bloodstream. The phosphate from the plasma that isn't bound to proteins, specifically about 90% of it, will be filtered by the glomerular capillaries. Next, in the proximal convoluted tubule, or in the PCT, 70% of the filtered phosphate is reabsorbed. This is done by a sodium phosphate cotransporter located in the proximal tubule cells. Okay, now phosphate actually has a so-called transport maximum, or TM for short. This means that at some point, the sodium phosphate cotransporters get saturated with phosphate. When this happens, the TM is reached and the remaining phosphate in the lumen is excreted. Okay, let's move on and talk about calcium. The 1% of calcium that's not in the bones is found in the ICF and the ECF, specifically in the plasma. Plasma calcium concentration is about 5 to 10 milliequivalents per liter, and it can be found in three forms. About 40% of plasma calcium is bound to proteins like albumin, 10% is bound to anions like phosphate or citrate, and the remaining 50% is the ionized or free form. Calcium is essential for muscle contraction, enzyme activity, and blood coagulation. It also helps with releasing neurotransmitters from neurons, as well as releasing hormones from the endocrine glands. Calcium also comes from our dairy products like milk, cheese, and yogurt, and the recommended daily intake is about 1 gram per day. Once ingested, it's absorbed into the bloodstream, and just like with phosphate, most of it goes to the bone. The rest circulates the bloodstream either freely or bound to plasma proteins or anions, such as phosphate or citrate, and some of it is excreted by the kidneys. So inside the kidneys, unbound plasma calcium is filtered by the glomerular capillaries into Bowman's capsule, and it enters the renal tubule. In the PCT, about 67% of the filtered calcium is reabsorbed, which is exactly the same percentage as sodium reabsorption in the PCT. This is not a coincidence, as studies suggest that calcium reabsorption may be secondary to sodium and water reabsorption in the PCT, but the exact mechanism is still not clear. As a result, if sodium reabsorption is inhibited, calcium reabsorption will be inhibited too, and when sodium reabsorption is high, like with dehydration, calcium reabsorption is increased as well. In the TAL, about 25% of the filtered calcium is reabsorbed, and it's also tightly coupled with sodium reabsorption. In the TAL, there's the sodium potassium chloride cotransporter, or NKCC2, which reabsorbs sodium, potassium, and chloride into the bloodstream. As such, they shuttle one sodium into the cell down its concentration gradient, and that powers the movement of one potassium and two chlorides into the cell as well. Now, the sodium-potassium ATPase pumps 3 sodium ions into the interstitial fluid in exchange for letting 2 potassium ions into the cell, in order to maintain the low sodium concentration inside the cell. Finally, both chloride and potassium move from the cell back into the lumen of the TAL through special channels on the apical side of the cells that simply leak these ions passively. The passive movement of potassium generates an electrochemical gradient, also called a lumen positive potential difference, that increases the reabsorption of calcium and magnesium through a paracellular pathway, meaning these ions don't use any channels, but rather they sneak between 2 epithelial cells and go back in the bloodstream. Finally, the distal tubule reabsorbs about 8% of the calcium, and here's where calcium reabsorption is finely tuned. In the distal tubule, calcium reabsorption is not coupled with sodium reabsorption, and it's regulated by a hormone called PTH, made by the parathyroid glands in the neck. In fact, both calcium and phosphate homeostasis are regulated by PTH, so when blood calcium levels decrease, that stimulates PTH secretion. PTH acts on 3 organs. In the bone, it activates osteoclasts, which are the cells that break down bone tissue, and as a result, calcium and hydrogen phosphate are released into the bloodstream. PTH also increases intestinal absorption of calcium. Finally, in the kidneys, PTH binds to a specialized receptor on the surface of the distal tubule cells, which triggers an intracellular signaling pathway that ends up increasing calcium reabsorption. To regulate phosphate excretion, PTH inhibits the sodium phosphate cotransporter in the PCT, decreasing phosphate reabsorption and increasing urinary excretion of phosphate, also called phosphaturia. Calcium homeostasis in particular is also regulated by 2 other molecules, calcitonin, which is produced by the parafollicular cells of the thyroid gland, and vitamin D. Calcitonin does the exact opposite of PTH, so it decreases plasma calcium levels and increases renal calcium excretion. Finally, vitamin D promotes calcium absorption in the gut, so it increases plasma levels of calcium. Finally, just a few words on magnesium. The 40% of magnesium that's not in the bones is distributed in muscles and other soft tissues. Only about 1% of magnesium, or a normal range of 1.4 to 2 milliequivalents per liter, is found in the plasma. Just like with calcium, the total plasma magnesium can be divided into 3 fractions. About 20% of magnesium is bound to proteins. Then there's magnesium bound to anions like phosphate and citrate and free ionized magnesium. Magnesium acts as a cofactor in a lot of enzymatic reactions. Some examples of reactions include glucose and fat breakdown, the production of antioxidants like glutathione, enzymes and proteins, synthesis of DNA and RNA, as well as regulation of cholesterol production. Because of its crucial role in enzymatic reactions, magnesium helps maintain good nerve and muscle function, including the cardiac muscle, so symptoms of magnesium deficiency include muscle spasms and cardiac arrhythmias. We get magnesium from foods like kale, avocado, and bananas, and the daily recommended intake is about 400 milligrams per day. From the GI tract, it gets into the bloodstream and circulates either bound or unbound to plasma proteins, and also some of it is excreted. Okay, let's see how the kidneys handle magnesium. About 80% of the magnesium from the plasma can be filtered by the glomerular capillaries, since 20% is bound to proteins and can't be filtered. In the PCT, about 30% of the filtered magnesium is reabsorbed. This is thought to be a passive process that's driven by sodium reabsorption. In the TAL, about 60% of the filtered magnesium is reabsorbed through a paracellular pathway. Alright, as a quick recap, phosphate, calcium, and magnesium are mostly found in the bone matrix. Calcium and phosphate make up the bone matrix and magnesium strengthens it. Phosphate plasma levels range between 2.5 and 4.5 milligrams per deciliter. Phosphate is mostly reabsorbed in the PCT using the sodium phosphate cotransporter. Calcium plasma levels are about 5 to 10 milliequivalents per liter. 60% of calcium is mostly reabsorbed in the PCT through a mechanism that's tightly coupled with sodium reabsorption, while 25% is reabsorbed in the TAL through a paracellular pathway. Both calcium and phosphate are regulated by PTH, which increases phosphate excretion and increases calcium reabsorption. Calcium is also regulated by calcitonin, which decreases calcium reabsorption, and vitamin D, that promotes calcium absorption in the gut. Magnesium plasma levels are about 1.4 to 2 milliequivalents per liter. About 30% of the filtered magnesium is reabsorbed in the PCT. 60% of it is reabsorbed in the TAL through a paracellular pathway, while in the distal tubule only 5% of magnesium is reabsorbed.
The blood calcium level stays stable thanks to 3 hormones: vitamin D, parathyroid hormone, and calcitonin. Let's focus on vitamin D, which along with parathyroid hormone helps increase calcium levels, whereas calcitonin helps lower them. The majority of extracellular calcium, which is the calcium in the blood in the interstitium, is split almost equally into calcium that's diffusible and calcium that's not diffusible. Diffusible calcium is small enough to diffuse across cell membranes, and there are 2 subcategories. The first is free ionized calcium, which is involved in all sorts of cellular processes like neuronal action potentials, contraction of skeletal, smooth, and cardiac muscle, hormone secretion, and blood coagulation, all of which are tightly regulated by enzymes and hormones. The second category is complex calcium, which is where the positively charged calcium is ionically linked to tiny negatively charged molecules like oxalate and phosphate, which are small anions, that are found in our blood. The complex calcium forms a molecule that's electrically neutral, but unlike free ionized calcium, it's not useful for cellular processes. Finally, there's the non-diffusible calcium, which is bound to large negatively charged proteins like albumin. The resulting protein-calcium complex is too large and charged to cross membranes, so the non-diffusible calcium is also uninvolved in cellular processes. Now, after parathyroid hormone, the metabolically active form of vitamin D, also called calcitriol, is the second most important hormone involved in regulating blood calcium. Vitamin D is a steroid hormone, which means that it's made from cholesterol and it's fat-soluble. Active vitamin D starts out as one of 2 metabolically inactive molecules, either vitamin D2 or ergocalciferol, which comes from plant sources in our diet, and vitamin D3, or cholecalciferol, which can either come from animal products in our diet or be made in skin cells that are exposed to sunlight. But since both ergocalciferol and cholecalciferol are physiologically inactive molecules to vitamin D, they have to be modified a bit by the body before they can be used. Let's start with molecules coming from our diet. When vitamin D2 and D3 reach the small intestine, they get packaged along with bile salts into micelles, which get absorbed into the intestinal cells called enterocytes. Vitamin D2 and D3 are then incorporated into lipoproteins, called chylomicrons, which get into the lymph and make their way through the lymphatic system and eventually enter our blood. Vitamin D2 and D3 are fat-soluble, so they have to be carried around the blood by vitamin D-binding proteins, which take them to the liver. Vitamin D2 and D3 get into the endoplasmic reticulum of the hepatocyte cells of the liver, and that's where they start to undergo multiple modifications. So let's zoom into the endoplasmic reticulum. First, the enzyme 25-hydroxylase adds a hydroxyl group in the 25th position of both molecules. As a result, vitamin D2 becomes 25-hydroxyergocalciferol, or ercalcidiol, and vitamin D3 becomes 25-hydroxycholecalciferol, or calcifediol. Ercalcidiol and calcifediol then re-enter the blood, once again bound to vitamin D binding protein. Their journey then continues from the liver to the proximal tubules of the kidneys. They enter the mitochondria of renal cells, and so let's zoom into the mitochondria. Here, the enzyme 1-alpha-hydroxylase adds a hydroxyl group to the carbon 1 position of both ercalcidiol and calcifediol, resulting in 1,25-dihydroxyergocalciferol, also called air-calcitriol, and in 1,25-dihydroxycholecalciferol, also called calcitriol. Both air-calcitriol and calcitriol are commonly called active vitamin D because they both have the same effect on the body's vitamin D receptors. Now let's follow the journey of vitamin D3 that's produced in the skin. Keratinocytes in the two deep layers of the epidermis, the stratum basale and stratum spinosum. And these produce 7-dehydrocholesterol, which is a precursor molecule for cholecalciferol. When the skin is exposed to sunlight, 7-dehydrocholesterol absorbs ultraviolet B radiation and is converted to pre-vitamin D3 via photolysis. Then heat generated by cellular metabolic activities causes the pre-vitamin D3 molecule to isomerize, or change its shape to form vitamin D3 or cholecalciferol. The vitamin D3 molecules then enter the blood, and follow the same path as dietary cholecalciferol to the liver, and then to the kidneys. Normally, total blood calcium is between 8.5 to 10 milligrams per deciliter. If calcium levels drop, the parathyroid glands release more parathyroid hormone, which has a number of effects. Parathyroid hormone increases bone resorption and releases calcium and phosphate ions into the blood. It also acts on the kidneys to increase calcium reabsorption in the loop of Henle, the distal convoluted tubule, and collecting ducts, while blocking phosphate reabsorption in the proximal tubule. Finally, parathyroid hormone also causes the renal cells in the proximal tubule to increase synthesis of 1-alpha hydroxylase, which converts calcifediol into active vitamin D. Decreased levels of phosphate can also trigger the increased synthesis of 1-alpha hydroxylase. Active vitamin D is then transported by a carrier protein through the blood to various tissues, like the bones, intestines, and kidneys. Because vitamin D is fat-soluble, it diffuses across cellular membranes of these tissues without the help of a transporter. Inside the cells, it binds to vitamin D receptors in the cytoplasm, which transport it into the nucleus where it stimulates transcription of specific genes and synthesis of proteins. In the bones, vitamin D functions very similarly to parathyroid hormone by binding to vitamin D receptors on osteoblasts, which are bone-building cells, and this gets them to release cytokines. These cytokines include receptor activator of nuclear factor kappa B ligand, or RANK-L for short, as well as macrophage colony stimulating factor, or MCSF. These cytokines help get multiple macrophage precursors to fuse together and form a single osteoclast, which are bone-eating cells. Osteoclasts break down bone, and the two minerals that make up bone, calcium and phosphate, are released into the blood. In enterocytes, which are the epithelial cells of the small intestine, active vitamin D increases calcium and phosphate absorption by stimulating the synthesis of calbindin D28K, a calcium binding protein that can hold 4 calcium ions, and it helps stimulate calcium absorption. When calcium from food sources diffuses into enterocytes, it binds to calbindin D28K before being pumped out through the cell's basolateral membrane and into the blood circulation. Active vitamin D also causes the enterocytes to increase synthesis of sodium phosphate cotransporters, which increase phosphate absorption. In the kidneys, active vitamin D carries out 2 similar actions. In principal cells of the distal convoluted tubules, it stimulates the synthesis of calbindin D28K, allowing more calcium to be reabsorbed from the urine. And in tubular cells of the proximal convoluted tubules, it causes increased synthesis of sodium phosphate cotransporters on the apical surface, which increase phosphate reabsorption. Alright, as a quick recap. Vitamin D2 is obtained from the diet, while vitamin D3 is obtained from the diet or the skin. Both vitamin D2 and D3 are transformed into active vitamin D by the liver and kidneys. Activation of vitamin D can be triggered by parathyroid hormone when calcium or phosphate levels are low. Effects of activated vitamin D include stimulating bone resorption, calcium and phosphate absorption in the small intestine, as well as calcium and phosphate reabsorption in the kidneys. Parathyroid hormone causes a net increase in calcium and a decrease in phosphate, while active vitamin D causes an increase in both calcium and phosphate.